Okay, so you have the PowerPoint, the original PowerPoint of this on your Google Classroom. So if you feel like you can just read through this and understand it, great. If you read through it and you're still like, I don't know how this works, I'm gonna kind of go through and just kind of explain this PowerPoint and just do an online lecture, just like I would in class, and kind of help clarify that. If you have any specific questions, if you could just comment below the assignment, that'll help me kind of gear our learning towards what we need. So the thing we're going to be talking about today is what we call protein synthesis. Now, it's the process of making proteins. So, if we can just kind of review what proteins make up all living material. It's going to make up hair, blood, nerves, muscles, things that deliver things in the cells, enzymes, antibodies, and what we call our cellular construction workers. It's what's going to be helping create things in the cells. So that's what proteins are. They're going to help make up everything that we do, including cells. So we're going to talk about how those are made today. So proteins are made of amino acids, and there's 20 of them. And I'll show you those 20 amino acids a little bit later. But different proteins are made by combining those 20 amino acids in different combinations. So let's say that I have 20 different ingredients. And I can take any of those 20 ingredients and I can make different things just by combining them together. Okay? So proteins are actually manufactured by the ribosome. So if you think back to our cell anatomy, here's our rough endoplasmic reticulum, okay? Here is our ribosomes, and the ribosomes are located on the rough ER, and these little black dots that are over there, okay? And then here is an example of what we call the protein making process happening in a cell, and we're gonna cover that in just a sec. So we've talked about what makes up protein. Let's talk about the function of them. One, they're going to help fight disease. They're going to help prevent you from getting the coronavirus, for example. Okay, that's what goes in a lot into helping figuring out why we get flu shots or what causes the flu or sickness like that. So that's what's going to help us fight disease. It's going to help build new body tissue. So let's say that you're a bodybuilder. Okay, it's going to help create more muscle tissue for you. It's going to help create enzymes, and enzymes are used for digestion and other chemical reactions that are proteins. So enzymes are going to speed up the rate of a reaction, and we'll talk about what enzymes do in just a sec. And it's a component of all cell membranes. Proteins help hold together that cell membrane, which helps hold together things like your skin tissue, heart tissue, liver tissue. So they're pretty important. And so these are all the things that are holding together your cells. We obviously want our cells to be held together. It's a pretty important thing. So these amino acids come from the food that we eat. They don't just come out of thin air. That's why we have to consume food. So things that are high in protein, things like milk, cheese, fish, any meats, steak, chicken, beans, those amino acids can come from the food we eat. And the proteins that we eat are broken down into individual amino acids that, again, we have 20 of them. They're just rearranged according to the needs and directions of what our DNA is telling our body to do. So let's say, for example, that you had some milk with your cereal this morning. Great job. Your body has enzymes in your stomach. Those enzymes are going to break down the protein that is in the milk, and that protein from the milk is going to be pushed out to your cells and pushed over to the ribosomes so that you can help create new protein cells and more bone and skin and hair tissue. So that's really why you have to eat food. So yeah, that's pretty much it. Okay. So protein synthesis works in a couple of different ways. So here's kind of the process that we're going to move through. First, you have your DNA. DNA is going to go through a process called transcription, okay? And that DNA is going to be transferred or turned into what we call mRNA or messenger RNA. That mRNA is going to go through a process called translation, which ultimately is going to take that and turn it into a protein. So let's talk through that process. So the first step is we're going to take that DNA and we're going to make proteins. So we're going to go through transcription. So making a protein. First, we have to make a copy of the DNA and turn our DNA into RNA. This is called transcription. So transcription is copying genetic information from DNA to RNA. There's a very specific thing that happens here. Why does this happen? So DNA actually has a genetic code for protein that needs to be made, but proteins are made by the ribosomes. Ribosomes are located outside of the nucleus where DNA is held. So that protein needs to be made, but proteins are made by the ribosomes, and ribosomes are outside of the nucleus, out in the cytoplasm. So since the DNA is too large to leave the nucleus, it's double-stranded, we have to turn it into one strand, single strand. We have to turn it into what we call RNA, ribosomal I'm thinking of the whole term, ribosomal NA, I forget the last two parts of NA, nucleic acid, that we're going to pull that RNA strand out, and we're going to take that over to the cytoplasm, and we're going to code it for proteins there. So part of that DNA, here's how this process works, and you can kind of see it start to happen here. We're unwinding that DNA strand, and part of the DNA is going to temporarily unzip and is used as a template to assemble complementary, what we call nucleotides, into mRNA. So if you remember the last time that we did DNA, we did our licorice lab. That was one of the things that we did. We're taking those nucleotides, adenine, thymine, guanosine, and thymine, ATCG, and you can see some of those matches here. And what we're going to do is we're going to send one strand out, and we're matching this RNA strand with one side of the DNA. And you'll notice some people are like, what's the U come from? The U comes from mRNA. So instead of matching up with thymine on mRNA or RNA, DNA, RNA does not have thymine. So A instead of T right here matches up with U. T is still going to match up with A. C is going to match up with G, CG, TA. But you'll notice when you have an A, the adenine is going to connect to something that we call uracil, okay? And the uracil is only on RNA, okay? So that's really the main difference here. And I want you, if you need to, you can stop and think about that, right? So RNA does not have thymine. It has uracil. So that's the only difference there. So if you need to, write it down for a sec and think about that. But that's the main difference, is that RNA only contains uracil instead of thymine. So you can see here that it only has A, G, C, and U that it's going to match up here. Okay, DNA is not going to change. DNA still has its own nucleotides here, but that's the main difference here. Okay, so we have this mRNA, AUCG, right, that's matched up with half of the DNA strand, and it's going to go through the pores of a nucleus. You can see it's kind of pulling itself out here with the DNA code, and that's going to attach to here, the ribosomes. So transcription happens in the nucleus, okay? And that process is going to happen here. So we're going to pull that DNA strand apart, okay? We're going to take an RNA strand and match it up with half of the DNA. We're going to take it outside of the cytoplasm, and that DNA code is going to start to attach to the ribosome. So that's going to take us to our next step, which is translation, okay? So now we're going to take that transcribed DNA or an RNA, and we're going to translate it into something that's readable. So we're going to decode that mRNA into a protein. This is called translation, and this happens in the cytoplasm. So transfer RNA is going to carry amino acids from the cytoplasm to the ribosome. So it's carrying it out, and it's connecting it to that ribosome, okay? And here's that happening here. So there's a series of 3 adjacent bases in an mRNA molecule that codes for a specific amino acid, okay? Remember, no T here, no thymine, only guanine, cytosine, adenine, and uracil. So each tRNA has 3 nucleotides that are complementary to the codon in mRNA. So each tRNA, transfer RNA, is going to connect to the mRNA on the outside, which is connected to the ribosomes. So that's going to code for a different amino acid. So here's your amino acid. It's connected to a codon. okay? And your codon is going to be in mRNA, and your tRNA is going to have an anti-codon. That's how I remember it, okay? So it's going to connect to those. So these three codons, I'm pointing at the screen like you can see, these three codons are going to connect to these three anticodons. Anticodons are connected to tRNA. Codons are connected to mRNA. So that should kind of help you remember it. So mRNA is carrying the DNA instructions, and tRNA is carrying amino acids to meet the ribosomes. Okay, so here's our messenger RNA. That's the one that we transcribe to the nucleus. The transfer RNA is happening outside. It's happening in the ribosomes. Okay, so the mRNA is going to enter the cytoplasm and attaches to a ribosome. Translation begins at what we call AUG. Okay, that's what's called the start codon. Okay, each transfer RNA has an anticodon whose bases are complementary to a codon. Anticodons match to codons, whose bases are complementary on the mRNA strand. The ribosome is going to position the start codon to attract its anticodon, which is part of the tRNA that binds it. Then the ribosome is going to bind the next codon, and so on. Okay? So then amino acids are going to be joined together to make a protein. Okay? So here's kind of an assembly line, and here's our ribosome. It's this whole little blue thing. It's going to just kind of pull down this strand, and it's going to connect my codons to my anticodons up here. Okay, so here's our transfer RNA connecting to our mRNA. And it's creating this polypeptide strand, which is a protein. So this is the translated part up here. Okay, this is all that's translated, and it's going to complete it for you. So here's the amino acids or the proteins that are being created. Okay, remember the start one, where did that one go? The start is AUG. So if I come down here, here's AUG. That's our start codon. Some of these I can't even pronounce, but you can see. And there's a couple of stop codons. So you'll see, here's our examples. Let's say that I have UCA. Well, UCA is what we call serine. Okay, serine can be coded in a couple of different ways. Proline can be coded in a couple of different ways. These are specific proteins that are going to do specific things in our cells. There's three stop codons, okay? UAA, UAG, and UGA. That tells the ribosome that we're stopping translation. Just says, hey, cut it out, okay? We're not doing that anymore. So that's when it knows to pull off and that we have to stop creating proteins. This process happens when you eat food. So this is the process that's going to happen when you have some chicken for lunch and you're breaking that process down. When you're done breaking down that chicken, then this other codon is going to come back, and it just tells your body we're not creating proteins anymore. Okay. There is a quick video on my slides if you'd like to watch. It's hyperlinked, so you can watch that video when you're done. I recommend doing that. And then let me know if you have any questions.